Hey everyone, we're here today. I'm here with Priscilla and Dr. Bonnie Maldonado, who's the chair of infectious disease for pediatrics at Stanford. Before that was earlier in her career was a fellow at the CDC, right around the time when HIV was really growing as an outbreak. And we have a lot of different topics that we want to cover. I want to start off by apologizing. We had an issue with the stream before, so we're restarting this, and hopefully it'll be a bit smoother this time. But I figured it's probably good to start off by going through some news that Bonnie had this week, which is that she's leading a team across Stanford and some folks at UCSF to do a pretty meaningful size test, a serology test and PCR test over a 9-month period to track the rates of infection and rates of exposure and how that evolves across the Bay Area to answer a number of questions that we don't know the answers to around how COVID spreads and how it behaves. So Bonnie, maybe you could just start off by telling us a little bit about the study that you're doing and what we hope to learn from it. And I guess maybe just to make sure that everyone kind of has a sense of the basic ideas here. I mean, maybe kick us off with explaining, just what a serology test is and a PCR test and all that.
Yeah. So we're going to be looking at PCR testing and serology testing. Basically, PCR testing is a way where you can actually look for an organism in a person's body. So in this case, you've been taking a swab, a nasal swab, and looking for evidence of that, the SARS-CoV-2 virus or the COVID disease virus in that swab. And the way you do it is you look for a gene. There's a test that's called PCR, and that test just takes a particular gene that's specific to that organism, and it multiplies it millions and millions of times in a machine so that you can detect that organism. And now we have that test available at Stanford and UCSF so that we can get a test result within 12 to 24 hours. So it's really rapid and it's very reliable. So that can tell you if you're infected with that virus right this minute. The other test is the serology. It's basically a blood test. And what that is it measures proteins called antibodies. And antibodies are proteins that your body makes that are directed against bacteria, viruses, anything that invades the body. And those antibodies can sometimes protect you if you see that disease again. So if you get sick now, you develop antibodies, you might be protected the next time you see that virus. And so we're going to be testing two populations. One is the general population that represents everybody in the 6 Bay Area regions that we've picked out: San Mateo, Santa Clara, San Francisco, Marin, Contra Costa, and Alameda. And we'll have a representative sample of people based on a lot of important demographic groups so that we can track how many people have COVID infection with the virus that causes COVID at the baseline, and also look for their antibodies to this disease. So hopefully we'll be able to find out right away how many people are infected and how many people have been infected. And since we know the virus hasn't been around that long, it will mean that they have been probably infected in the last 2 to 3 months. And then what the exciting part too, we'll be able to follow those people over time at once a month so that we can see if there are new infections happening over time in the whole Bay Area, so that as we go through the next few months with Governor Newsom's and the public health department's plans to change, get us back to kind of a normal situation, we can track and see what's the baseline rate of infection now, and is it going up or staying the same, and hopefully going down over time. And if there are hot spots, we'll be able to identify those in real time because the results are back very quickly, and we can give those results to the county health departments. So that's the one test study. And the other one will be a sub-study among healthcare workers that is very similar, except we're also going to be focusing on some very special antibody tests to understand the nature of those antibodies. Are they protective? Do they protect you from infection? Do they protect you from disease? That is, you could become infected but not get sick. And we can hopefully be able to distinguish between those two. And then finally, if you get antibodies and they are protective, how long will they last? Because that might be important to know for the next time the virus comes back, if it does.
Yep. And with these tests, there's a lot of talk around, we see all different numbers reported. So I'd love to, I read that New York has 20% seropositivity. Like, what is that? Are there any practical implications of what that means for New York? And
Yes.
Can you talk a little bit about false positives and false negatives here?
Sure. Well, before we talk about that, let me explain to you why false positives are important. And they're really more important at a population level, because basically what they mean is you test somebody, it would be like saying you're testing somebody for the presence of antibodies, for example. And there is always going to be a little bit of error in some of these tests, for a lot of error. And what we're finding is that there's a lot of error in some tests. Now, we know that Stanford and UCSF tests are very, very accurate. So very few false positives and false negatives. But let me just give you an example. If you had 100 people who you wanted to test, and you knew that 50% of the people were infected, and you knew that the test you used had a 5% false positive rate, that would mean that if you tested those 100 people, you'd get 55 positives. 50 of them would be real positives, and 5 of them would be false positives. Now, you don't know which ones they are. You just know that's the way the test works. So that's 5 out of 55 people. So about 10% of the people would actually be false positives. So that's not great. But let's just say the number of people in your population actually only has 5% infection. And we think that's where we are with COVID right now in the US as a whole. That would mean that if you had 5% infected, that 5 people would test positive and it would be really infected, and another 5 would test positive because the test has a 5% false positive rate. And that means that 50% of your test results are false positives. So the false positive rate doesn't change, but the proportion of population changes. And that's why you need a really accurate test, because if you're not using a very accurate test, then you're gonna overrepresent the number of infected people in a population. And that's a problem, because you might underestimate the number of deaths. So for example, if you say, well, look, 50% of the people are infected and yet we only have X number of deaths, that must mean the number of people dying relative to the number infected is very low. And so you want all those things to be as accurate as possible. And that's why our tests are very accurate. And not only that, if we get positives, either by PCR or by the blood test, we're going to actually trade samples with each other, so we can run the test again with the other UCSF and Stanford's test. So having the test done by 2 places by 2 different methods will even further ensure that we're not seeing false positives.
Yes, there's a lot of questions that people have about what does having antibodies mean? And there are a lot of questions that still need to get answered. If you have antibodies, does that prevent you from getting the disease again? How long do the antibodies last for if they provide some kind of immunity? Do they prevent you from spreading it to other people? How are you planning on using the studies that you're running to help answer those questions?
Yeah, so we're expecting that most people are not going to ever have a positive PCR or a positive antibody test. That's why we need to look at so many people. We need to test about 7,500 people a month, same people over time, because we think the number of positives is going to be low. But among those positives, we'll be able to track over time whether they get reinfected. For example, if somebody has an antibody that's positive, will they later on, if let's just say the virus comes back or it starts to have a seasonality to it, we can see if people who have antibodies are less likely to be infected again than people who don't have antibodies. And we'll also be able to see how long the antibodies last. And then finally, in the laboratories of our scientists at Stanford and UCSF, they're going to do some other tests with the actual virus itself to see if they can determine how well the virus protects protects from getting the infection into human cells in a laboratory setting. So this could pave the way, among others, for looking at how a vaccine might work down the line.
Interesting. Well, I mean, you asked this question a second ago about what do the population level exposure numbers mean? We've seen some of the numbers coming out of New York that serology tests there have shown that up to 20% of people might have been exposed. And there's a preprint that our team flagged coming out of Italy in Lombardy showing that potentially 60% of the population in Lombardy has been exposed. What do those numbers mean for public health, and how would understanding that level of exposure inform the type of public health decisions that governments and local officials need to make?
Well, the very first thing it tells you is what's the degree of protection, what's the degree of protection you need to have what we call herd immunity, right? So we use vaccines, for example, to give people immunity to diseases so that when there is a random case of an infection, there's so many people who are protected that you basically only have 1 or 2 cases, and then there's nobody else will get infected and the disease stops spreading. The reason we're seeing so much COVID is because nobody has ever seen this virus before. So it's just spreading from one person to the other. The only way you can stop that is to have people who have immunity or to keep people apart. And so with this particular virus, given the transmissibility, we think that you need to have at least 60 to 80% of people with immunity in order to stop it from spreading. We don't want 60 to 80% of people in the US or the world to get this disease so we can stop it from spreading. So that means we have to do other things like keeping people apart. And so that's why that number is so important. And we know that, for example, in Lombardy, where there was a particularly big outbreak, and maybe in parts of New York, there may be higher rates of infection. I know that a very high rate in one German town was about 15%. So 60% sounds extremely high to me, but that might have been in a very small community, for example, where lots of people were exposed to one another very early. But I would say that in the US, it would be very unusual for us to see more than, say, a 5% infection rate across the US right now, which makes it really scary to think, well, you need 60% of us to be immune, to have immunity in order to stop this from spreading all the time. So that's why a vaccine is going to be really, really important, or drugs that people can take to reduce the amount of virus that is replicating in their body. So to keep them from, yeah, go ahead.
Well, I was just going to take a moment to remind as pediatricians, COVID is not the only thing that we're trying to protect the community from, and that we have existing vaccines in which we've reached herd immunity levels through vaccinations. And you had raised this point earlier.
Yeah, absolutely. So we have to, I'm a pediatrician as well, and I know that a lot of people, we have seen record numbers of under-vaccinated kids in the last month or two. And I understand why people are afraid to go outside with their kids, or afraid to, maybe they might be afraid to go to their doctor's office. And in fact, the American Academy of Pediatrics has suggested that we really focus on the first 2 years of life rather than the older kids, and just wait a few months until the older kids can come back for their well-child visits. But you need to make sure that those young babies get their normal vaccines, because we still have other viruses and bacteria around that can infect children. So we want to make sure that that people come back, they talk to their doctors, they get their vaccines. It's really what pediatricians do best. So I agree.
Yeah,
we don't want to have a we don't want to have a.
We don't lose the herd immunity on the other diseases that we've already done this hard work.
Exactly.
Is there anything else that on kids that you think is not particularly well understood by people at this point about how the disease works? And I mean, it's one of the fortunate things about it seems to be that kids don't get the worst symptoms as much. But what are the big questions that are still open, and what do you want to make sure that people know about this?
Well, I have to say that unfortunately, we know almost nothing about this disease in children. I think part of it is because we've been so overwhelmed with taking care of the adults. But nobody, and we were afraid to have, schools closed down very quickly because as we talked about, we needed to socially distance. So we haven't really had a chance to look at what happens to children. As far as we understand so far around the world, it doesn't look like kids have a very high rate of infection, or else they don't seem to have a very high rate of disease. That is, they might be getting infected, it's possible, but they don't seem to have a lot of symptoms. Now, that's all very anecdotal, and there have been very few studies to date in children. So we are doing some studies ourselves here at Stanford. I'm doing a study looking at household transmission to see what's going on with kids and adults as well. But there's a lot we need to understand. And I think one of the key things that we could understand about kids that could help everybody is if children really aren't getting infected, why is that? Because almost every disease that we know of really affects children, that is widespread. Usually it affects children first because they're outside, they're putting dirt in their mouths, they're not washing their hands, they can't control their secretions very well, and they're all huddled together usually. So they're very good at spreading infectious diseases. Why are they not getting sick? We don't know the answer to that. That might be helpful. Or the other question would be, let's just say they are getting infected, but they're not getting sick. That would also be important to know, because then we could figure out, well, if they're not getting symptoms, maybe we could use that same mechanism to prevent older people from not getting symptoms as well. So there's a lot we can learn from children, and we've been trying. But right now, most of the federal agencies are really focusing, and rightly so, on just the people who are getting hospitalized and put on ventilators, and those are mostly older adults. We do know one thing. We do know that kids we're seeing some evidence of inflammatory reactions in children. So things like, we know that young adults we're starting to see strokes in young adults. And we're also seeing that kids are getting these funny rashes and some fevers and other symptoms. So we're not really.
When you say young adult?
In their 20s to 40s. So yeah, in the age range where we don't think people are getting that sick. So they're not getting that sick, but then later on, they seem to be having later onset complications.
Got it. Yeah, that's one of the big open questions is, to the extent that people survive, do they have long-term damage and to different organs or different things? And that I guess will need to be studied over time. So you've studied a lot of different diseases, right? I mean, you spent a long time working on polio and some of the work around vaccines there. I'm curious what experiences that you've had either fighting polio or fighting HIV that you think are transferable or could teach us lessons about this fight against COVID?
Well, let me start with the social aspects, right? So the social piece of it is, I'm actually I'm surprised that even in this day and age, we're seeing a lot of fear and anxiety. So I know that I've seen from other countries, hopefully not so much here, but people are afraid of healthcare workers. They've been, I've heard of reports of healthcare workers being harassed or beat up because they're afraid that they're going to be spreading the disease. And that's actually unfortunate because those are our first responders. And I remember the HIV days when people were afraid to treat patients who had HIV. I used to have to go every day and give talks to the nurses and the doctors and explain to them that all of the evidence we had suggested that these diseases were not easily transmitted to people unless they had exposure to blood or blood products, et cetera. And I feel like that might be happening a little bit today. So I wouldn't say that the fear and anxiety is at the same level, but it's a generalized fear. Everybody's afraid. And I think we need to be cautious. I do think we need to be careful, but I think we should not be afraid of other people. And so that's one of the things I've learned. The other thing I've learned is that this virus is very similar in that the most symptomatic people are probably going to be a very small proportion of the whole group of people. So this is going to be like a pyramid. I think what we're seeing is the tip of the iceberg or the tip of the pyramid. And there's going to be a lot of people who are probably not getting symptoms. Now, the only good news here for us compared to HIV is that HIV took 10 or 12 years before you could actually get really, really sick. And this disease is moving much faster. So we should be able to understand more about the baseline of people. And then if we can understand that, we have a better shot at treating those people for their infections so that they don't spread it to others. That's very similar to the polio story, where most people with polio don't get sick at all, and they're actually spreading the disease to other people. And in that case, when that happens, you need to vaccinate everybody because you just can't pick up everybody who's infected.
How will we know if you have that? We talked about serologies. How will we know if you have in fact have their antibodies, whether or not it actually stops you from passing it on to someone else, like in polio?
Well, unfortunately, the only way you're going to know, you could do some lab tests. So for example, you could take the virus, mix it with antibodies, and then try to see if it infects cells. And if the antibodies prevent the cells from getting infected, then you know, at least in test tubes, that it seems to be protective. But ultimately, you just have to give it to people and see if they are more or less likely to get infected. So it's going to be risky. I think you're going to have really good lab evidence that it works in the laboratory and maybe even in animal models. So for example, the very early study you might have heard about from, I think, is it earlier this week? Somebody told me they're called blurs days now. I can't remember what day it was, but it may have been yesterday or the day before. Don't know. Anyway, there was a day sometimes recently where the UK scientists had a vaccine against a virus that's related. It's called MERS, or Middle Eastern Respiratory Syndrome. It's another coronavirus. They had already been working on that vaccine, so they were able to retool it very quickly so that they could put in some of the genes for proteins from this virus, from the SARS-CoV-2 virus. And they tested it they had a lot of data on safety already from the other experiments. So they were able to put this vaccine virus into 6 rhesus monkeys, and those monkeys were actually fully protected from getting sick from the SARS-CoV-2. So now they're rapidly moving into more animal and human studies. So they're hoping that they can scale it up very quickly. And so I think it will be interesting to see if you do animal studies where an animal has a very similar disease outcome as we do. If that works really well, then we can start using it in people.
We have a question here from Han. From Taryn. When you create a vaccine for a virus, doesn't a virus change? How do you actually create a vaccine to something that's always changing?
That's a great question. So RNA and DNA viruses mutate all the time. It turns out that RNA viruses mutate faster than DNA viruses for they're just the way they're built. But this RNA virus has a special, unusual mechanism that allows it to, for some reason, it doesn't mutate that as quickly as other RNA viruses. So we know that this virus mutates more slowly just from studying over 3,000 to 4,000 samples of these viruses over time and looking at their genetic sequences. And we know that they haven't been spreading over time or mutating over time. And so we've been very fortunate that way. We think it'll be very similar, but even other viruses, like for example, measles virus, we think measles virus has mutated a bit over many, many years, but yet the same vaccine that we used in the 50s and 60s is still effective against measles that we see today. So there are parts of the virus that never really change or change very little. And that's generally where the vaccines are directed, against the parts that are conserved the most highly and less likely to mutate. But that's true. Theoretically, you could have a virus that eventually mutates away. That's why we have to invent a new flu vaccine every year.
Yeah. One thing that's interesting is that we've talked about with Joe DeRisi's lab is that you can track these mutations, and it's sort of like a genetic fingerprint and traces where these viral strains are coming from. And I think that's something important to see, that it's coming from all over the world. And we see different strains coming from Europe and Asia and different parts of the United States. And it's related to that fear idea that you were talking about that was so prevalent in, my point is related to the fear that was so present in HIV. I think a lot of people are, fear is triggering stereotypes and different acts of racism. And I think the science tells us that actually the coronavirus is coming from all over the world.
Yes.
Is that your experience in looking at the next strain data as well?
Yeah, and I think that's the issue. We, in my field, again, as you mentioned, I trained at CDC, and I've been working in global health for almost all of my career, as well as in the US. And it was just a matter of time. It would have come from anywhere. I think, putting people together with animals that we've never seen before and changing climates, all of these things, you roll them together and you're going to have, random mutations occur at the wrong time, and you can see something like this. Look what happened with Zika, for example. That, you don't want to really, you learn one thing, is that we're a global community now. If we weren't global 100 years ago, we certainly are now. And I think we're all on this planet together. So, and, I don't think anybody would purposely, despite what we've heard, nobody's purposely made this virus. So these things just happen. We have, for example, the US is one of the places where we see lots of antibiotic resistance, because we use antibiotics all the time that make our bacteria mutate. So we shouldn't be blaming other people for things that just happen because of our own, that we might be doing as well in terms of breeding these resistance. What we need to do is approach these things scientifically and learn how to undo them. So that's a great point.
Vaccine development for a second. I'm curious if you could give folks a sense of what you think that they should expect, right? I mean, there's a lot of timelines that are thrown out around this will be ready and around the middle of 2021. As someone who's been involved in vaccine development, and the complexities and the different types of vaccines that someone might try to create here, what are your expectations of what would happen, and what should people overall, how should they be thinking about this process?
Yeah, so vaccines take a long time. I mean, on average, a brand new vaccine that you just start from scratch. Now, remember, this vaccine already had a backbone built a long time ago. It takes about 10 years, and it takes, hundreds of millions of dollars to build out a vaccine, because you want to make sure, first of all, that it's safe. And secondly, that it actually works, and it works well. And sometimes we're willing to sacrifice effectiveness. That is, it might not work 100% of the time. No vaccine does, but it may not, some vaccines may work 30% of the time. But at this point, if it worked 30% of the time, I think people would be willing to take it. But you need to make sure it's safe. And so that's really one of the issues that I think we need to think about here is where are we along that pipeline? And I know there are over 40 vaccine candidates that are out there right now, but most of them aren't even ready to be put into animals yet, much less humans. And those studies take a while, because most of that is about safety. You want to make sure that they don't have negative effects that would be worse probably than the disease, than COVID. So those things generally take a while, and the UK vaccine shows a lot of promise. And I think if they're going to share that platform, I'm sure they would share so that other people can start looking at other vaccines that would be very similar and building those out rapidly. So I do think that at least a year will go by before we have anything that we think is safe enough to put into people. But in the meantime, I think there are a lot of other things we can do besides distancing, which is obviously important. But that would be looking at repurposed drugs, so other drugs that we have that are available that might work. It's interesting that this drug, the remdesivir, it works just pretty similarly to the way the original HIV drug worked. So there's only so many mechanisms that a virus may respond to. We understand the way this virus works pretty well. So the idea would be to identify drugs that we think will work against different pathways that the virus takes to attach itself to a human cell, to replicate in the human cell and then to come out and infect other cells. So there are many pathways along the way where drugs could target those. And then you could use those drug targets as effective ways to prevent disease. Again, coming back to your example of HIV, we still don't have an HIV vaccine, but we think we can control the disease just by using therapeutics. So that may be a way to get us through until there is a vaccine available.
Maybe you can say a little bit more about remdesivir. Like, is everyone going to get it now if they're positive for coronavirus, or who is it best used for, and how much does it help?
Yeah, well, so the current trials were really done very rapidly, which is great, but they were very focused. So these were done in people who were already hospitalized and either had moderate or severe disease. And so the trials showed that that people who got remdesivir compared to placebo, meaning a sugared pill or just no drug at all, but they thought they might they had a 50-50 chance of getting something. Those trials showed that the people who got the remdesivir were significantly less likely to progress and have more serious disease, and they were also more likely to get better over a shorter period of time. And there was a trend towards decreased mortality as well in those patients. The other study that showed that that trial, the one that showed that decreased time to getting better, was a study of 5 days versus 10 days. It was a 10-day study of drug. And then there was a separate study looking at 5 versus 10 days of drug in severe disease. And in that study, they demonstrated that 5 days was just as good. But it is an IV drug. So the question is, really, what are you going to be? Yeah, you might be able to use it on the outpatient setting, but mostly right now what the FDA is doing is they have said it should be used in people who are hospitalized with severe disease. And the good thing is they said children or adults. So children could get it as well. Yes. Mm-hmm.
Yeah, so it's a drug that can't be taken by mouth. It has to be given through.
Right. Well, and then I'm sure there will be efforts to see how else you can formulate the drug and make it better, or make it available on an outpatient setting, or do other studies to look at outpatients or less people with less severe illness. But it's the first step to understanding that in fact something does work, because that gives people hope, and that gives scientists a lot of information to go to the next set of drugs. We're already doing two drug studies here at Stanford, one on the outpatient side and another one on the inpatient side. And so, and I know other people are doing similar studies.
Yeah. I think we only have a few more minutes, so I'm curious if there's anything else that you want to make sure that we hit that we haven't so far.
I want to make sure people know that this is a really life-changing moment, and if everyone feels like this is not normal, you're right, it isn't normal. This is a once-in-a-century event. But I think back about what happened with Ebola. I worked in the DR Congo during the Ebola epidemic. I didn't work on that. I worked on something else. I worked in sub-Saharan Africa on HIV when 30% of pregnant women that I was working with had HIV disease. And we have made remarkable progress. And I want everyone to remember that this is something that we can all get over. We need to just stick together and have hope, and whatever it takes to get you back into a Zen kind of place, you should do, as long as you're following the rules. Just feel hopeful. I'm an optimist. I'm a pediatrician, so I guess that's part of being a pediatrician. I have hope for the future, and I think about it. We haven't really known about this virus for more than 4 months, and we've already got a vaccine that looks like it could work, a drug that might work. It took us so much longer for disease like HIV. So I think we're going to be looking at some real progress in the next few months, and pretty soon we'll be able to get outdoors, and you can still get outdoors and get some fresh air and exercise. Just keep your spirits up, and trust good information sources like this.
Yeah. Thank you. The work that you're doing and others in your community, it gives me hope. So I really appreciate it. We all do.
Thank you.
Thank you for your
All right.
leadership. We're looking forward to following the study, and we're proud to support you in the work that you're doing.
Well, thank you. We're very excited, and thank you so much.
All right.
All right.
Thank you.
Great.
See you soon.
All right. Have a good weekend.